Okay, you're welcome. I'm happy to be here. Thanks for the invitation. I have the chat window up, so if anybody wants to raise their hand or whatever you call it in WebEx, feel free to do that, and just we can make this an interactive discussion. Jenny set me up with a few problems that you guys have around, especially around curating variants from older papers for which transcripts may not be available, or at least were not specified. So I thought I'd create a really kind of broad-based set of slides. It'll be pretty short, and then we can turn this into a discussion about specific problems you have, and I'll see what I can do to help you out with those. So by way of background, as Jenny said, I'm on what's called the Sequence Variant Description Committee, but it's really the committee that oversees the standard for HGVS. I got into this because I was an early member of Locus Development, which eventually became Invitae. And as part of that, I was involved in lots of software development for our curation efforts, and especially about how we did, I think, exactly what you're doing, identifying papers either in clinical reports or papers, and then trying to curate information associated with those. And we came across genes that we could not offer as on a panel test because the current tools were not good enough to support those. And I'll dig into that a little bit in a minute. But really, this was by necessity. We need to build a pipeline that allowed us to curate variants in genes that had some diabolical issues, and so we needed to build a tool to do that because the then available tools were insufficient. So I'll describe some of that in a minute. Again, feel free to interrupt if you have any questions, and let's dive in here. Next slide, please. Okay, so forgive me, I don't know where everybody's starting, so I'm going to start. I won't do too many prefatory notes here, but HGVS obviously is a standard through which we represent variation on all sequence types, and I think that's important. So it's unlike VCF in that HGVS can support all sequence types, that is genomic, transcript, and protein sequences, and kind of implicitly RNA. It is kind of the standard. It's was originally supported by the Human Genome Variation Society, and after sort of getting shoveled around, is back with the HCBS after the Human Variome Project closed down. The standard is required by many journals and clinical labs. It is kind of the de facto standard. I'm hard-pressed to think of any other standard used to represent variation for communication any purposes. And obviously, it looks a little bit like this simplified variant shown on the slide, where there's a reference sequence, a indication of the sequence type, and then a location and a description of the change. As if any of you have used HCBS, obviously this is as simple as it gets, and there are quite hairy possibilities of possible with HCBS as well. But we won't spend too much time with those unless you have questions about those. Next, please. So the HGVS, I think, especially from a curator's point of view, I think comes to play with when we have to project or transfer a variant that's discovered in genomic coordinates into a variant that is represented in either transcript or protein coordinates. And I think this is probably the place that most people really start using HGVS. That is, we identify variants in genomic coordinates during sequencing, and in order to understand the impact of those variants and interpret them and communicate them, we have to translate them into transcript and protein coordinates. And conversely, once they're published and then read by people, we take those transcript or protein coordinates and try to infer where those are in the genome. I call this process projection. That is, we project a variant from one sequence to another, and we'll get into technically what has to go into that, what information you need around in order to do that, because I think that's at the heart of the question that, about how to interpret historical variants. All right, next, please. Okay, so I happen, as I mentioned at Invitae, I led the team that developed our HCBS manipulation tools. We then open source those tools, and it's become that package, also called HGVS, is become one of the more popular packages for manipulating HCBS expressions. And it does a bunch of things. I break down the operations that it does, especially the ones that are most user visible, into kind of three basic areas. So there's validation, which is given an HCBS expression, do I even know that it's valid? Are the coordinates within the bounds of the transcript? Does the reference residue that's specified in the variant match with actually the reference residue on the reference sequence, and so forth? There's another operation called normalization and rewriting, which are slightly different things. Normalization, as many of you will know, is the process of representing a indel, del-int, by right shifting it. And it's tempting to think you could do that by eye easily, but there are some diabolical cases. It gets challenging pretty quickly, and so the package we wrote provides a way to do normalization. In a strict theoretical sense, it's really hard to write it's hard to be precise about what normalization actually means, but in this case, it means shifting a variant through a repeat region as far right as it can be represented, and so the package we wrote provides that. Then there's also rewriting, which is slightly different. That's primarily around writing an int as a dupe, or perhaps an inversion when that's a del-int as an inversion. Those kinds of changes are rewriting. They don't change the nature of the variant, but they provide a sort of human interpretation on what actually occurred at that sequence. Importantly, both of those operations, validation and the normalization components, requires a database of sequences. In order to normalize or validate a sequence, you of course have to have the underlying sequence. And that is one of the challenges in building tools like this, because as soon as you build a tool, and especially if it's open for transcripts of all forms, suddenly you might get any sequence, passed through you might get any variant, a variant with any transcript comes through that tool that then needs access to the underlying sequence, and especially if you care about performance, those sequences really need to be local. So that's a problem that we've solved, but I think from a curator point of view, it might be worth just keeping in the back of your mind that every time you do something like normalize a variant, it really requires that you have the underlying sequence available. And then finally, the projection is, as I said, transferring a variant from one sequence to another, and the challenge there is even more difficult in a way than just having a large number of sequences available. It requires having a database of transcripts, including those exon structures and the alignments of those transcripts to the genome. And this is where historical interpretation of variants really becomes challenging. NCBI doesn't provide a historical alignment of old transcripts to old genomes. Or more correctly, what they do provide is an alignment of the most current transcript to a current assembly. And so if you read in a paper an older transcript, you are a little bit hard pressed to ever find the alignment that refers to that, that is necessary to interpret that variant. And so in order to solve that problem, we wrote something called the Universal Transcript Archive, which is a little bit what it sounds like. It more or less monitors NCBI and keeps an archive of the alignments over time. and does that not just for NCBI, but also stores ensemble transcripts as well, so that we understand when an ensemble transcript is truly equivalent to a RefSeq transcript and when it's not. And by equivalent there, I mean, same exon structure, same exon coordinates, same CDS sequence, which turns out in some situations doesn't have to be the case. And so UTA is what we use to keep all the information around. And some people have picked that up. That's also freely available, and some people have picked that up to use for other operations as well. Okay, next slide, please. Okay, so I think probably a lot of you know about tools, especially things like Mutilizer. Just to highlight the ones that I've been involved with, obviously, therefore, a little bit the ones that I'd recommend are the ones at the top here. So Variant Validator is a user interface, a lot like Mutilizer in functionality, provided by actually another group, University of Leicester. Raymond Douglas's group has put that together. That's built on top of HCBS, Seq Repo, and UTA, which I just told you about. So if you're looking for another tool to use, which is probably no one ever is, you can go try Variant Validator. It is, again, it's roughly equivalent to Mutilizer. The other one that I highly recommend, a commercial tool, is Alamut, which probably maybe many of you have used. I really think that's great. I've followed that for many years. Aside from being a commercial tool, it's actually really great. And they are pretty diligent about handling some of the more challenging cases in HGVS manipulation as well. Next slide, please. And by the way, omission of talking about SNP-F and VEP was not a slight. That's just for time. Those are also great packages, which is why they're on that list. So the package we have is that we provide is this HGVS package. It's completely open source. It's got a very liberal license. You can use it for anything you want, basically, including commercial tools if you want. It's got code from 15 contributors from 8 organizations. It's got a really extensive test suite. When we first did this in 2014, or at least published it, probably 2/3 of the authors on the paper were actually from Invitae's curation team. They contributed a bunch of tests from hand curating variants and projecting those from one sequence to another. And that, I think, is actually one of the things that makes the package really solid, is that it had lots of input from curators in construction. It's really easy to install. It's extremely easy to install, especially given the complexity of what's involved. You can install just the package, and by default, it will use remote sequence and data sources on the fly. And the only cost of that is that is a kind of performance penalty for having to go across the network. It's also quite easy to install locally, which makes it much faster. And obviously, if you're if you're using this for clinical purposes on patients, I highly recommend that you install it locally just for privacy reasons. And maybe what I think of as kind of the most important feature of the HGVS package is really that it handles difficult cases very well. And hello.
Give me a second. Sorry, can everyone put themselves on mute, please? Thank you.
And so it'll take a little while to go into some of what makes manipulating HGVS difficult. We can either do that here. There's also, just this week, a paper we wrote on that shows some of the cases where HGVS does well, and especially in comparison to some of the other packages, is there on the screen. And so you can go read that if you want to see some of the diabolical cases that I think the HGVS package handles particularly well. Next, please. Okay, since Jenny brought it up, there is a problem that I'm sure many of you come across where you read a paper, find a variant, and the variant doesn't specify the transcript. And this is a perfect kind of technical slide to just show you that actually there is a tool in the HGVS package to do exactly this. Right now it's just a command line script, and I think that probably doesn't appeal to many of you. But the functionality is there, and I think with not much effort we could figure out how to get that into, I could talk to the variant validator folks and make that likely get that into a user interface. And so it looks a little bit like this. So the first one is a variant in HFE, right, 2 that's specified with just a gene symbol. And what you see on that next single line that I've kind of wrapped into 3 lines is that for the original input, there are 5 plausible variants, each filled in with the transcript that is a valid transcript given that variant. So all that's going on here is that, given a symbol, we can look up all the transcripts. We try the specified sequence change on that transcript and then call the validator to ask the validator whether this looks valid. And if it does, then we return it, and if it doesn't, we don't return it. So in the next 2 cases, you can see that that we specify TNF receptor 1A variant. I've just pulled this from dbSNP, so you can see what dbSNP thinks on the right. 123 T to C. There's only 1 transcript for which that is a valid, looks like a valid HGVS expression, and so only 1 is returned, despite there being several transcripts available. And it works for RNA variants as well. All right, next. This is variant validator. Just wanted to remind you that this exists. Next. And then now it's my turn to listen. I'd love to hear what problems you guys face, and if the HGVS tool does what you want, I'm happy to point you to that. And if it doesn't, then maybe we can figure out, how difficult it would be to actually implement something that would be useful to you. And that's it.
Great, thank you so much. Does anyone have any questions? Any cases that have been particularly difficult? Because I think one of the issues, as you mentioned, Dr. Hart, that we often come across are these older papers where, we just don't have the transcript, or there's just not a huge amount of information available. Sometimes you'll be lucky to have some sequence information in there that you can actually map it back to a transcript or gene and figure out what's going on. Sometimes you just don't have that luxury. It just gives you, 187 C to T or something like that. So, I guess each case is different. You just kind of have to use the tools available. But, do you have any general thoughts on that? These look like good tools. We also have an allele registry through ClinGen as well that can help with some of this. But just any guidance or general guidance or thoughts on some of that?
Well, it depends a little on how old the transcript, how old the paper is. If you are looking for a RefSeq transcript, I think there's a reasonable chance that it's in UTA, especially if it was out in the last 5 years or so. There are relatively large number of transcripts that are more difficult to find. So a lot of the old BIC transcripts are difficult to find. It turns out those are in UTA too, but those required some manual reconstruction, by me, where I manually aligned those to the genome and used a bunch of papers to figure out where those were valid and ran those by curators. So the BIC transcripts happen to exist too, but I suspect there's a lot of transcripts out there that I just don't know about that were used in kind of smaller communities. And I think the essential element to get across is that really, if you want to represent if you want to project variants between sequences, you have to have the alignment between those sequences. And I think one of the mistakes that's often made is that the alignment doesn't have is a trivial alignment, and that and specifically the assumption is that the transcript sequence agrees with the genome. And that's a really tenuous assumption. It is wrong often enough that I would not rely on that. And so maybe I should just pause there for a minute. Is that obvious to people that a transcript sequence is not the same as the reference sequence, will not agree with the reference sequence? If that's not common, maybe that's something we should talk about.
Yeah, why don't you go ahead and just give us some information on that? I think that would be helpful.
Okay, so then maybe Jenny, can you go to slide? I think it'll be slide 14. Right, back up one, please. Yeah, there. Okay. Right. So transcript sequences, have been, cDNA sequenced sequences have been around a lot longer than even the human genome sequence in many cases, and they come from different people. So there's no reason. In fact, there's a lot of reason to think that they wouldn't agree in general, right? Because they come from different people and different populations even. So what that means is that if you take a transcript sequence and align it to the genome, and specifically I should probably say a ref-seq sequence, and align it to the genome, any assembly, it will, in the vast majority of cases, agree perfectly. But in a fair number of cases will not agree. And sometimes those disagreements are due to polymorphisms. In fact, probably the most common case is a polymorphism and a single SNP polymorphism. In some cases, that polymorphism can be an indel, like what's on this page, on this slide. And in a small number of cases, they actually represent sequencing errors, most often actually in the genome. In fact, that was one of the cases that really caused us trouble. We wanted our panel to include NEFL, and I remember NEFL included the gene NEFL, and the panel and that genomic sequence actually has a sequencing error in the middle of the CDS, and in fact that would not be a viable person likely as a sequence. Poly G track with 7 Gs, and there should only be 6, and that obviously throws the ORF out of frame. So that kind of problem that kind of shows up and really wreaks havoc when you try to project variance between the sequences. And so to drill into that just a little bit more, imagine that you've got, sorry I can't use a pointer here, but imagine that you're on at the transcript T just before that gap, right? If my counting's right, that'll be C.25, yeah, right there. So that'll be C.25. The next position, of course, will be C.26, but notice that doesn't correspond to anything in the genomic sequence, right? And so it's only when you get to C.29 is there a correspondence again. So you can imagine that if you are unaware that this gap exists and you naively step from 25 to 26, from the T and G, right, at the start of the gap, if you didn't know that gap exists, you would incorrectly assume that aligned with the GG on the genomic side of that. Yeah, exactly, right there. And so this is where this is a place where HCBS does the right thing. We do enough bookkeeping that we know where these gaps are. Thanks to UTA, thanks to the alignments kept in UTA, we know where these gaps are, and so we can use those during projection. Utilizer does a lot of things really well, but this is a place where it gets this wrong. So if you are, and it gets it wrong systematically in every single case where there's a gap in the transcript. If you go to Mutilizer and enter a variant that is before a gap like this, you'll get the right answer. As soon as you step across the gap, Mutilizer doesn't know about these gaps and makes the false assumption I just mentioned, and will get the coordinate wrong. And so that's something to be aware of as you're using Mutilizer, that this will be wrong systematically if you have one of these gaps, and it's particularly hard in Mutilizer because you don't know whether you're in such a transcript or not. So, so the point of this slide again was that that the transcript doesn't agree with the genome. In cases where that causes an indel, that creates a reference frame glitch, that makes projecting variants from one to the other more difficult, and HGVS gets this right. And so this is an example where you really need the alignment, and it's in order to do the projection, and the alignment's got to be right, otherwise you get the wrong answer when you do this projection. Anybody want to ask any questions about that? Does that makes enough sense?
Yeah, I think that was really helpful.
Okay.
So, Reece, this is Ronak here, and does HGVS provide recommendation regarding such kind of variants and projections?
Yes, it does. So it tries to do, if you are not in the vicinity of this, then the bookkeeping is right and extremely well tested. So if you are clear of this area, then yes, it gets the right answer. The cases, as you might imagine, it's intellectually it's a little bit challenging to go through all the cases. We do in the paper, and so you can read that if you like. But there are a lot of cases here. So imagine you specify a variant that's right in the middle of the gap. Well, what is the right answer when you project that to the genome? And I think there are at least two possibilities. One is there is no such variant there, so you don't write anything, which would be G dot equals or something equivalent to that. Or conversely, you decide to project that whole gap, the whole missing region, into the genome and then make the variant change. I hope that made sense. But it'd be the equivalent of taking that GGA and dropping it into the genomic sequence as though it existed, and then making whatever variant change. And that's, in fact, what HGVS does. And the reason we do that is because that's the way you guarantee that this is a reversible change. That is, if you take that same genomic variant then and project it back to the transcript sequence, you get this what I think of as a nice reversibility property, that you get the same transcript back out when you project the other direction. There are also other variations on this theme where you have a variant that spans this entire region. Then what are you supposed to do? And so it gets a little thorny. But so I think the short answer to your question is HCBS mostly handles these for you, and so it comes up with what I think is the right answer, or at least a justifiable answer. That's not to say there aren't any problems. I'm sure there are, but we have lots of tests around this kind of stuff. Any other questions?
I have a completely unrelated question, if you don't
All
mind.
right. Okay, no.
One of the genes I work on has a lot of cryptic splice sites, and I think I understand the use of the parentheses inside the P dot for the amino acid change, but I feel like the rationale, we should be using the parentheses inside the P dot most of the time. And I guess I'm just confused. I think we assume with most missense variants that that we do understand that would be the amino acid change, but that it probably hasn't been proven. But we still put the parentheses on the outside of the P. Am I making sense?
Yeah. So I think what you're asking is really about is just the use of parentheses in general, irrespective of the cryptic splice sites, right?
Yeah, I mean, it seems like most of the time no one's really done the experiment to say that truly is what's going to happen, but that's the purpose of putting the parentheses inside the P dot, right, is to show that this is what we think is going on, but we don't know yet. And with the cryptic splice sites, I think it's easy to know, yes, we think this is what it's going to look like, but we don't really know. But I'm just wondering, are we misusing it with most of the missense variants? Like, are we not doing the nomenclature correctly?
So this is a place where I actually differ with Johan a bit on, who is the kind of godfather of HGVS. I wish we didn't use parentheses. So I'll give you, I'll tell you what the official line is, and then I'll tell you my opinion.
I have to say, this is Deb. I agree with that too. That the parentheses has been really hard to integrate. But please go on.
Yeah. So the intention, as you said correctly, is that parentheses indicate something you infer, not something you. So my problem with that is a couple-fold, and I think it's a place where HGVS in general maybe oversteps. I think anything that, to me, that looks like annotation or explanation that's layered into the syntax of an expression is a bad idea. You know, we don't, by putting the parentheses around, you're saying, you're trying to hint to somebody that you're not really sure that this is right or not. And I think those kinds of hints need to be much more explicit. So that's just one. I think, you know, there's all kinds of annotation you wouldn't put into a variant, right? If in a protein sequence, you wouldn't put that a tyrosine's phosphorylated, or, you know, whatever. Like there's all kinds of things you could imagine putting in there, but we don't. And we don't because it's just, it tries to overload and strain the syntax too much. The other problem with it is that there's actually lots of other places where I think in HCBS where things are inferred, you know, including a variant that's projected onto a transcript from a genomic sequence. One could argue that there's, you know, in complicated cases, there's going to be editing or other processes that make that change untrue as well, and so it's also kind of its own sort of inference. So basically, I think it's too much. Practically speaking, it also creates the problem that people invariably end up comparing with and without parentheses. And from a computer's point of view, if you've got parentheses there, it's not equal to the same, not equal to the thing that doesn't have the parentheses. And so I think it creates confusion that's really not terribly helpful. So that's my opinion about it. And in HGVS, in the package HGVS, we actually have a flag that you can set that is to turn off parentheses. So by default, it does the standard thing, which is to add the parentheses, but it's really easy to turn that off. And that came in large part because I polled our clinical geneticists, and like everyone to a T said, no way will I use those. And so, you know, from a practical standpoint at Invitae, like, we were not going to use the parentheses. And so that's how we ended up not using the parentheses. Does that answer your question?
Well, I totally get why you guys don't use parentheses, but I think for standardization right now, the guidelines recommend using them, right? So I guess I'm just sort of at a loss of when to put them inside, when to put them outside the page.
So, yeah, I think this is it. I think in this particular point, I think the HGVS standard and especially Johan's insistence on having them is, to be honest, at odds with what much of the de facto community does, you know, what the community does de facto. It's people, I actually have a hard time thinking about any group that uses them for reporting. I don't know, that's maybe wrong, but I can't think of a report that I've seen recently that has, you know, a clinical report from any lab that has parentheses around them.
Yeah, well, I'll give you an example. So ClinGen has a special issue in human mutation coming out, and human mutation demands that those recommendations are followed.
Yeah.
Right. We've had to go back through in parentheses all of ours because we didn't have that as entry. And I guess the issue we have, or I have with the parentheses is, that's an annotation. So it's not have I personally tested it, is there evidence in the literature that this has been tested? So you have to go back through literature and look to see if there's functional tests for that, about whether you put parentheses on or not. And I agree, that's an annotation rather than a nomenclature, I guess. A question I have with that would be, there is no way, correct, that you put parentheses on the transcript, right? Where you could say, I'm not certain of which transcript I'm using.
That's right. There is that's right. There's no such equivalent annotation on transcripts.
Okay.
I have a question, Reece. So you mentioned that that the Python tool is fast and in mapping and projection and normalization. So what is what do you mean by fast? Like per second or per minute? How much it processes?
Well, sorry, I might have said faster when you have local. I can answer the question, but I didn't mean to make any performance assertions there. But I can tell you that with local installation, you can pretty easily hit 50 projections a second or more. That includes a full validation pass, which is expensive computationally. But that's by default, HGVS tries to be fairly rigorous, and so and kind of pedantic about generating variance. So when all of the checks are on, you can process about 50 variants a second or so locally.
Yeah, I was just wondering what will be the case of whole genome sequencing, say for example 3 million variants.
Right. Yeah, I don't know the answer to that off the top of my head, but obviously could be a test we could do. You're thinking about annotating a VCF, for example, or something like that?
Just for example, like if I want to look into the converting, say for example, VCF file into HGVS expression, and just maintaining it, how fast or how slow it is. Like that was the question.
Yeah. So I don't think I've ever done that test on a full, on a whole genome sequence, VCF. There is a tool in the package. It's a kind of unsupported experimental. It's in a directory called experimental, but it does exactly what you said. It rewrites, it takes a VCF file in, spits a VCF file out, and adds HCBS expressions to the info field. So, I think we could probably test your question pretty readily. My guess is, though, it would take a while. Obviously, 3 million variants is a lot to go through. So on the kind of back of the envelope, 3 million divided by 50 a second would be my answer, which is kind of an unappealing answer, but that's probably what it is.
Yes. And the recommendation that you provided for software tools, are they based on some kind of taste or just personal experience, or
well,
community?
We can go back to that maybe and go through those in detail. Jenny, can you back up to whatever that is, maybe slide 6 or so, please? Yeah, right there. So, obviously I'm going to be biased toward tools that I've built. I think the paper that I mentioned earlier, I think, is a pretty good justification for why I think the HCBS package is preferable to at least to Mutilizer. And that's, first and foremost about the correctness. Getting the right answer mattered to me a lot, mattered to the team. So, I think that's what I think about most. There's also HGVS is built by a group of people, so it's got a community of support around it. It's open source, and you can install it locally or remotely. All of those things I just said are not true of the Mutilizer package, and I don't mean to take anything away. Mutilizer did a lot of really good things for the community, but it's that correctness bit is one that I'm particularly hung up on, and that we at Invitae were particularly hung up on. And so I think as a recommendation, I'd have to go with HCBS and be kind of careful about Mutilizer in cases where the variance, or especially when their variants are in transcripts that have genome transcript discrepancies that will throw off the register of projecting variants back and forth. For the other tools, they do slightly different things. So, Alum is an amazing visual tool. So if you want to visualize where variants are, and it's terrific. SNPF does a nice job of annotation. Some things I don't agree with, and some of the HGVS expressions it generates are actually incorrect, but mostly it adds a lot. And VEP I have a lot of respect for. I think that's built all in the Ensemble pipeline. It's a tightly integrated tool, and I think works pretty well. And we used it for a long time at Invitae. So I think the answer to your question is, I guess, for just the processes that I mentioned earlier about validation, normalization, and projection, I would definitely go with HCBS. I think it's the best of the tools. It doesn't do lots of other things that the other tools do. So it depends, if you need to visualize, for instance, HGVS is not an option. Hopefully that answers.
Yes. Thank you very much for the answer.
So I'm curious about some of your extra slides too, and were there other things that you thought would be helpful for us to know? Unless anyone has other questions.
Yeah. So, well, sure. Why don't we? Maybe just go there, and we can sort of flip through a couple of them. There are some suggestions. Let's see. So, right, genome transcript discrepancies I mentioned. One of the things that is probably worth knowing that was a big shock to me 6 years ago or something when I got into this. So RefSeq transcripts, the authoritative source for RefSeq transcripts, of course, are at NCBI. The UCSC then takes those and realigns those to the genome. And the problem with that is that, and they use a different tool to align them to the genome. And so the problem with that is that the coordinates actually differ. So for a relatively small number, specifically, I think it was 886 transcripts about 4 years ago, had significantly different coordinates between UCSC and NCBI. So the problem with that is that it sets up the likelihood, not just possibility, but actually likelihood in those transcripts that a coding variant is interpreted as non-coding or vice versa when projected from the genome to the transcript. And that's obviously a problem if you're trying to infer effect from, say, an exome panel or something, or a whole genome sequence for that matter. So, so the caution there is just be aware that if you come across a variant and you're looking in the genome browser, or you think it might have been originally reported using genome browser coordinates, it's worth comparing to what RefSeq says, what NCBI says in their genome viewer as well. I consider the NCBI genome viewer to be the authoritative source of exon coordinates, and in fact have pretty good evidence that the coordinates from NCBI are more correct on average than those from UCSC. So there's one, another kind of note. Personal suggestion, I guess. You're the curator, so I want to be careful not to tell you exactly what to do. But I do notice that annotation is sometimes associated with what I think of as kind of the wrong variant, and I think that's mostly because the bioinformatics tools are not available to do what I think is probably the more correct thing. So imagine an annotation that should be really on a protein sequence, but for whatever reason is attached to a genomic sequence. There are a couple problems with that. So so for instance, if you had like a catalytic serine or something, right? That's annotated. That clearly should be an annotation in the protein sequence. A not uncommon challenge is that annotation, because of the way systems are sometimes designed, gets pushed to the genomic sequence. And so what you see is a position in the genome is associated with a catalytic serine, or whatever. You can. The point is that you're kind of projecting across these sequences, and there are a couple challenges with that. So the first is that obviously, especially with after transcription and translation, there will actually be multiple nucleotides associated with a single amino acid position. And so if you're really concerned about annotating functionality around an amino acid, you should put it there and not in the genome. The other problem is, though, that if for genes that have multiple transcripts, what do you do with a variation and annotation on the genome that is projected to that is intended only in the context of one transcript, but now you kind of have to know enough, you have to know a lot more to know that that annotation only applies to some of the transcripts that are relevant at that region of the genome, and maybe not all of them. And so probably enough said. Just, I think having the right tools so that you associate the annotation with what you're actually curating, I think is worth thinking about. I think I love the Ensembl process, and I love lots about where, how Ensembl works. With that said, and they do a great job on genome annotation and transcript annotation. With that said, clearly, especially in the United States, RefSeq is far and away the most common, and I think it makes everybody's life easier if one uses RefSeq transcripts and not ENSTs or others. That's not a technical discussion, it's more a practical suggestion. Another one is that I think maybe is not widely understood is RefSeq sequences actually don't contain intronic sequences, and that's relevant. When we talk about validation, for instance, if you have a HCBS expression that says something like C.20 plus 13, right? Where the plus indicates that you're 13 nucleotides into an intron, you can't actually validate that, right? So if you say 20 plus 13 A to T, you can't validate that because the RefSeq sequence actually doesn't have that intronic sequence. So the best you could do is project that to the genome and then validate the sequence there. But the reason that's a bad idea is that between different assemblies, you might have different underlying sequences. So the short thing to remember is you can't actually validate what's happened inside an intron with a RefSeq sequence without making some other assumptions. And this one came up just on the HGVS website recently. There was a kind of long discussion, including a published paper that got a bunch of these, made some assertions about variants being wrong when actually because they had made some of these assumptions but not written them down about which about trying to validate variants within intronic sequences. Okay, so let's see. Let's skip. What else? Yeah, the slides that I put in the backup, I think are ones from other talks that I thought might come up in questions. So there are no significant points to raise, but maybe go to 20, and we'll kind of close there with a fun kind of challenge.
Let me know which. I
think maybe one more.
One more?
Yeah.
There we
go. Sorry. Yeah, right there. Okay, so this is a hypothetical circumstance that I suspect exists, but it's hard to prove, and it is a problem with the way normalization, with the way I think some groups probably implement normalization. So Normalization is the process, as I said, of in this case, right shifting. Normalization is a process of moving a variant to its rightmost position in the case that there are multiple, equivalent ways to write a variant. So let's imagine that there's a variant in indel between 999 and 1000 here on the genomic sequence at position 1. And then if you naively apply normalization, that could shift to position 2. So if like an int A at 1 would shift to its rightmost position, would become an int A within the CDS. So now imagine that you then project that variant down to a transcript on the minus strand, and that first becomes 3, but then at position 3, but then you right shift that to position 4, and imagine that that's what you end up reporting in a journal somewhere. And then a curator comes along, picks up the variant that's reported at 4, and says, okay, I'm going to project. I wonder where that is in the genome. I'm going to project that up to 5. And then I'm going to right shift it because that's what you do. And suddenly you're at 6. So in this funny, by applying normalization just to the sequence without thinking about exon structure, we've effectively hyperspaced our variant from position 1 to position 6. And so the caution that I want to throw out to you is that these tools, I think we're at a point where we're kind of discovering some of the impacts of rules that we've made about how to operate, and how to, we should right shift variants, we should, and how to project them up to the genome and so forth. But I think if applied too naively, there's that can create problems in reporting variants. This case is actually why the HGVS package by default does not normalize variants across intron-exon boundaries. So it would specifically in the 3 to 4 it would not normalize across that boundary unless you explicitly tell it to. So I think this is just a fun case to consider about kind of where things, how things go awry when everybody's even trying to do the right thing. Things still end up in kind of can end up in a funny place. And I don't have any particular advice about this other than I think it's good for us all to kind of be aware of the kinds of challenges that can show up even when people are trying to do the right things with reporting variants.
Also, for this particular variant,
yeah.
Wouldn't it be difficult to say that there is no, the mutation in the genome can lead to exon or I think inclusion of intron, right? It can, right? It is difficult to say confidently that it transcript also it is going to be substitution. It can be large inclusion or deletion based upon how it affects the transcription, right?
I think your question is it's difficult to know how this would affect splicing, right? How a particular variant here would affect splicing. Is that effectively?
Yes. Yes.
Yeah, I think that's right. It's I'm not intending to make any statement about the impact on the biological impact, but certainly the reporting consequence of this is is pretty challenging.
That's great. Any other questions? We just have about 5 or so minutes left. I think this has been really helpful just to think about some of the different issues. Sorry, did someone have a question?
Yeah, so for your tool, that Python tool, is there a maximum number of variation, like say for example 10,000 base pair, or that it supports, or it can handle any length of insertion deletions kind of?
Yeah, that's a good question. I'm not sure I've ever tested that explicitly. There are no hard-coded limits. So I think it would be limited by by memory or something else. I don't know. That's a good question. I should go throw it some really tough large insertions and see what happens if I try to create a DEL-INS with a megabase.
So many a time during ClinGen curation, at least we came up with 2-3 situations where we needed to work with variation that when you projected onto genomic space, it is more than 10,000 base pair, but it is reported in literature and it has some annotations associated with it.
Yeah, interesting. Well, yeah, I'll go try that experiment and see what happens. My guess is it'll work just fine, but I'll go try it.
Oh, and speaking of which, are the examples on the HGVS website meant to work in the allele registry?
That's a good question. I don't know. I've never tried that, and I'm not directly associated with the allele registry, so I don't know that. But I would hope they would. On the other hand,
sorry, we have Ronak on the call. Ronak, do you know that question?
I think when we developed the allele registry, we used documentation provided by the HGVS as a base of our implementation. And I'm not sure, but if there are some specific example, we would have tried, but I cannot answer that exactly right now. Like whether we tried those example or we did not try. But I think we built the implementation based on the documentation.
Okay. Yeah, I asked the question based on what you had just mentioned about the large exon deletions, because like one of the first examples for how to write exon deletions on the HGVS website, that one doesn't actually work in the allele registry because it says the reference is wonky, but it's just kind of hard to figure out how to write these things and put them into a allele registry when the examples given on their website don't work for us to pull up to know, yes, this is the right way we should be putting it in there.
Yeah, I see what you are trying to address. Probably I think we should get in touch with someone. Probably Reece can help us get in touch with HGVS and can help to put example of exon inclusion or exon deletion if it is not there on the